Welcome to AP Biology 2.8, Tonicity and Osmoregulation. This is Dr. Joseph. In this chapter, we are going to study osmosis, which is the movement of water across a semi-permeable membrane. And we are also going to study tonicities, what isotonic, hypotonic, and hypertonic mean. And finally, we are going to look at water potential and look at an example on how to calculate it. So, osmosis is diffusion of water. Okay. The main idea is if there are more water molecules present on one side of a membrane compared to the other, water molecules will diffuse freely across that membrane until there is an equal concentration of water on both sides of the semi-permeable membrane. That's osmosis. Whereas in diffusion, again, we can think of osmosis as a special case of diffusion and where water is moving. But here you can see essentially that water is a solvent and water as a solvent is moving across the membrane. So here you see an experiment where you have a container with water, less water molecules on this side and more water molecules on the more water molecules on this side and less water molecules on this side because of the presence of a solute, and which means that there is less quantities of water on this side, therefore water keeps moving until there is the same concentration of solutes on both sides of the semi-permeable membrane. So the word tonicity means the capability of a solution to modify the volume of cells by altering their water content. Okay, so what is the capacity to move water basically? Okay, so here again I'm going back to this experiment. You can see when you have a higher solute concentration and you have less water on this side, in other words less free water on this side and more free water on this side where there are less solutes. What does that mean? For example, if you take sodium chloride, the crystal sodium chloride, and you drop it in water, it is going to dissolve, meaning it is going to become Na plus and Cl minus ions. And water is a polar molecule and becomes attracted to ions. So in other words, the ions are going to be surrounded by water molecules. And what that means is free water molecules are going to become less. Free means not bounded to the Cl or the Na+. So they are going to become less in quantity. That's what's happening here. Here let's assume that we added sodium chloride. What that meant is that sodium and chloride, Na plus and Cl minus ions, have attracted the water, and therefore there is less water on this side, and therefore more free water on this side, and more free water rushes into this to the side where there is less free water. So when we are talking about tonicities, or in other words the ability of a solution to move water, there are three types of tonicities that we discuss. One is hypertonic, the other one is isotonic, and the other one is hypotonic. What we need to remember is that when we talk about tonicities, we always talk in relation to something else. Like if I take a solution and I add water and there are more solutes in that solution compared to the cell that I put inside, then that solution is hypertonic, remember, hypertonic, which means more solutes compared to the cell. And if I have a solution that has less solutes compared to the cell that I added, then that solution is hypotonic to the cell. If there is equal amounts of solute both inside the cell and in the solution, then the solution is isotonic to the cell. So when a cell is isotonic to the solution, it means there is no net movement. It is not that water is not moving, but water is moving from inside to the outside in the same rate as water is moving from the outside to the inside. So there is no net water movement. The cell remains stable. In hypertonic solutions, when I'm putting a cell in a hypertonic solution, it means that the solution has more salt content and therefore less free water compared to the cell, which means that water from inside the cell is going to keep flowing out until the amount of solutes inside the cell is the same as the amount of The concentration are not the same, but not the amount, but the concentration of solutes inside the cell is the same as the concentration of cells in the solution that is keeping it. Therefore, it will keep on, water will keep on coming out, causing the cell to shrink. And in hypotonic situations, because there is more solute inside the cell compared to the solution which has less solute, which is therefore hypotonic, that hypotonic solution means that there is less more water, free water here, so that keeps rushing into the cell, and that cell expands and can eventually burst, which is called cytolysis. So in our class we did a potato core experiment, and you did some measurements of potatoes that were kept in different concentrations of sucrose over a period of 48 hours. And so that's kind of a representation of the experiment. We have a cylinder of potato core. We drop it into a beaker, either with pure water or a dilute sugar solution or a concentrated sugar solution, and we observe it after a period of time. Now, when it is, you can imagine that the potato cells are not pure water inside. Potato cells are going to have some amount of solute inside, some amount of sucrose inside. So what that is going to do is, what that means is the potato core is now placed in pure water, which is a hypotonic solution. So as you can see, hypotonic solutions means the cells are going to expand because water is going to start flowing inside them. Therefore, the potato core is going to increase in size. When it is an isotonic solution, let's say it is a dilute sugar solution, and the amount of sugar in the solution here is the same as what is inside the cell, water is going to move in and out in the same rate. Therefore, the potato core is not going to expand or shrink in size. Whereas if I put it in a very concentrated solution of sugar, what that means is there is less free water available in that hypertonic solution. So water is going to flow from inside the potato cells to the outside, causing the cells to shrink. So we weighed all of these potato cores, and based on, when you find the situation where there is absolutely no increase in weight, when you find that concentration of solution where the potato did not increase in weight at all or did not decrease in weight at all, that is the isotonic solution. So based on that, you can say the potato core's sugar concentration or glucose concentration is the same as the solution outside. Okay. This is just another way of doing this experiment, a dialysis tube. So dialysis tube is what is a semi-permeable membrane, allows some things to go through and not the others. Small molecules can pass through. So sugar can, so here we have sugar solution in the dialysis bag, and we are suspending it in a beaker of water, and over a period of time you can see that water is going to keep flowing inside because sugar cannot escape, but water can move freely. So water is going to keep moving inside, and then eventually the concentration is going to be the same inside and out. But in that process, because of all the water entering, the dialysis tube expands out. Okay, what about plant cells? Plant cells have cell walls in addition to the cell membrane. The thing is, cell walls are freely permeable. They are not semi-permeable. So now a plant cell has a freely permeable cell wall and a partially permeable or a semi-permeable cell membrane. What that does is, water keeps going in. For example, if I put it in a solution that has, that is hypertonic, and so a solution that is isotonic. So water is going to keep moving freely through the cell wall into the plasma membrane and out again through the plasma membrane. So the cell is going to be firm. The cell wall is going to be pushed against because of the cell membrane, and in that case a healthy plant is turgid. But if I place that cell wall, if I place that cell in a solution that is hypertonic, so what that means is water is going to escape out. But remember, nothing is going to happen to the cell wall because the cell wall is is sturdy and it has, it's not going to change shape, but the cell membrane is semi-permeable. So what that means is water is going to escape. In that process, the cell membrane is going to be pulled off the cell wall, and that is called as plasmolysis. And plasmolysis, here's a picture of how a plasmolyzed cell is going to look like, and these cells usually die. Okay, so the third topic here is water potential. Water potential is the ability of water to move. Ability of water to move, in other words, ability of water to do some work. It is a potential of water. It is represented by the Greek letter psi. It looks like a little trident. So think of Poseidon with a trident. So that's psi. And the water potential being is measured either in bars or bars or pascals or megapascals. There are two factors that influence the water potential in a living system. One is called as the solute potential, the other one is called as pressure potential. Solute potential depends upon the amount of solute dissolved in the system. So, for example, this is a U-shaped tube, and in this U-tube that's separated by a semi-permeable membrane here, water, if it's long as it's pure water, is going to be at the same level on both sides. If it's just pure water on both sides. But imagine if I add a lot of sodium chloride to one side. What that means is there is less free water here. So the water is going to flow from this side, from the left side, into the right side until the concentration of pure water is same on both sides. So as a result, the water level is going to rise. Another way you can change the height of the water from one side to the other side is by applying pressure. You can apply either positive pressure or you can apply negative pressure, and that's going to change the height of the water inside the tube. So solute concentrations, or the presence of solute, and the application of pressure are the two things that influence water potential, the ability of water to move. So it is represented by the formula psi is equal to psi s plus psi p. Psi s is the solute potential, and psi p is the physical pressure, or the pressure potential that is applied there. In pure water, there is no solute potential. Because there is no solute, the value of psi s is going to be 0. Okay, and here's one more term to remember. In plant cells, the force that is applied by the plasma membrane on the cell wall is called as turgor pressure. Okay, when it is not pure water, whenever we are adding solute, the solute potential is going to be not 0. It is going to be less. So that's represented by this formula minus ICRT, negative ICRT. I refers to the concentration, I refers to the ionization potential. So basically the number of ions the solute can break up into. If I add glucose is not going to break up into anything, right? It is just going to be 1. Sucrose is not going to break up into ions, so it is still going to be 1. If I add sodium chloride is going to break up into Na plus and Cl minus. So the ionic potential of sodium chloride is going to be 2. Okay, so usually in a problem they will give you the values for the ionization potential of the solute that they are using. If sodium chloride is going to be 2, if it's sucrose, it's going to be 1. C refers to the concentration of solute that in the solution. So how much solute was added, and that's the molar concentration of the solute. R is a pressure constant, and it's a constant value. It's 0.0831 bars per mole per Kelvin, liter bars per mole per Kelvin. And that value again will be given. You don't need to memorize this. And T is temperature presented in Kelvin. Remember that 0 degree Celsius is equal to 273 Kelvin. If I have 10 degrees Celsius, you just add that 10 to that Kelvin value. So it's 10 degrees Celsius is going to be 283 Kelvin. 273 plus 10. Same way 20 degrees Celsius is going to be 293 Kelvin. Okay, remember that that this is a negative value, so you have to have minus ICRT. So I would like for you to use this information to solve a problem here. Okay, so the value of psi in root tissue was found to be minus 4.3 bars. If you take the root tissue and place it in 0.15 molar of solution of sucrose at 20 degrees Celsius in an open beaker, what is the psi of the solution and which direction would the net flow of water be? Okay, here's the thing. When they are talking about open beakers, the psi p, the pressure potential, is going to be 0. Okay, that's one thing you need to remember. And also remember that water potential moves from a place of high water potential to low water potential always. So students get confused when we are talking about values in the negative value. So for example, you might end up with the pressure potential inside the cell as minus 2.3, and the pressure potential, I'm sorry, not pressure, let me rephrase that. You might see the water potential inside the cell as minus 2.3 and the water potential outside the cell, the solution in which it is placed, as minus 4.3. And the question will be, what is the direction of flow of water? So the question is, which is bigger? Is it minus 2.3 or minus 4.3? The answer is minus 2.3 is bigger than minus 4.3, right? Because it's a negative value. So water is going to flow from inside the cell, which is minus 2.3, to the outside, which is minus 4.3. In that process, what is going to happen? Your cell is going to become shrunken, right? It's going to shrivel up. So there's one more problem here. Again, remember, when you're saying there are two particles, the ionization potential is going to be 2. So when you're calculating minus ICRT, the I value is going to be minus 2 for sodium chloride. Okay, so I hope you're able to work these questions and find some find answers. Okay, so that was the summary of chapter 2.8, Osmosis, Tonicity and Osmoregulation. And hopefully by now you're able to understand what osmosis is and what the different types of tonicities are: isotonic, hypotonic and hypertonic, and also what is water potential, the formula that is used, and the factors that influence water potential, which are pressure potential and solute potential, and how to calculate solute potential, and how to use the formulae to understand water potential in a given problem setting. Okay, thank you very much.